Can you see me? So we're going to do some experiments today. So I'm going to do some interesting different live streaming. It won't just be me sitting there. So what we're going to learn today is how to purify plasmid DNA with all the materials that were sent with your Bioengineering 102 class. Now, the one step we won't show is growing up the bacteria. So for that, what you're going to have to do is you're going to have to make up a tube of LB broth. LB broth is 20 grams per liter. So if you're making up a 10 ml tube of LB broth, that will be 100 times less, right? Yes, 100 times less, which is 0.2 grams. So 0.2 grams for 10 mLs. Put that on your heat mat or in an incubator, possibly shaking if you can, and let that grow overnight, 16 to 18 hours at 37 C. And you should get a cloudy solution. It should look something like this. Cloudy solution. Now, once you do that part, then you're ready for the experiment. Now, the first thing that you're going to need to do is get a beer. So make sure you get a beer and you're ready to do that. Now, once you have your beer, then you're ready for anything. Hey, Kent, how's it going, man? Glad you joined us. So, plasmid DNA that we're going to purify today, plasmid DNA is basically the workhorse of genetic engineering. If you're trying to do any genetic engineering, you're trying to do it with plasmid DNA. So what you need to do, what everybody does, is they grow up that plasmid DNA in bacteria because it replicates it so well, right? So we have the bacteria that have grown up our plasmid DNA. You're going to grow up 2 tubes, one for GFP plasmid and one for RFP plasmid. And you're going to purify it. In this demonstration, we're just going to do one. So we want to get this plasmid out of the bacteria. That's the first goal. But we need to get the bacteria. So that's what the centrifuge is for. So we got a centrifuge here. Now, what we're going to do is we are going to take this 10 ml and we're going to put it in some microcentrifuge tubes. If you have a bigger centrifuge, a 15 ml centrifuge, you could spin down the whole 15 ml tube, but in this case we don't. Now you want to use all the bacteria in the tube. Our centrifuge is 6 place, so that means we could use 6 tubes. So let's fill all these tubes up equally. So a centrifuge, you want it to be balanced. By balanced, what that means is all the tubes across from each other, because it spins in a circle, need to be the same weight. Right? So you want to make sure all the tubes that we're using are approximately equal weight. Now this doesn't have to be exact, but it really depends how fast you're spinning. The faster you spin, the more exact you need to be. Alright, so we put it all in these tubes. Now we'll close up these tubes. Alright, so we'll go over to the centrifuge here. See, the centrifuge has 6 different holes. Now what I was talking about balancing, you can never spin a centrifuge with just one tube in it. What'll happen is it'll be out of balance and things will go crazy. So what you want to do is you always want to place a tube directly across from it that weighs the same amount. Right? Always in a centrifuge. So you can't do 3 tubes either. You need to place a tube across that weighs the same amount. Actually, you can do 3 tubes in this centrifuge. You have to balance it circularly, so it would have to be like that. But in general, you're not going to do 3 tubes. So what we're going to do is we're going to put all the tubes in here. Now, in a lot of these guides, in mini prep guides, they say you need to spin it at a certain wavelength, at a certain RPM. That is not completely true. What you have to do is spin it until it does the thing you want to do. In this case, we want to pellet cells. So you just have to spin it until you pellet these cells. And generally that takes about 2 to 3 minutes. We'll see. You'll see the tubes form a pellet. Oh, I didn't plug in my centrifuge. Give me a second. Here we go. The centrifuge is spinning. Now we just got to give it some time to let this stuff pellet. Now, I'm going to use this, hold my pipe tips. Okay, so now the first thing that we need to do is we need to bust open the cells and we need to degrade any RNA. Now, RNA has really similar characteristics to DNA, so if we're purifying DNA, generally we're going to purify RNA with it unless we get rid of the RNA somehow, right? And a way to do that is to use enzymes that only want to cut up RNA. In this case, we use RNase. RNase is added to the A1 buffer. So here's the A1 buffer. No, Mark, it's not possible to overspin it. You can generally never over-centrifuge stuff. Just spin it until it's clear. Keep spinning. It generally, whenever you're spinning stuff, it's not possible to over. Usually it's possible to under centrifuge, but rarely over. So the first buffer we're going to use is this buffer A1. We added RNase to it for you. Now, it says store 4C, but RNase is pretty stable, so you don't have to worry about that too much. Now, what this RNase will do is it will degrade, hopefully, the RNA in our sample, so we'll only purify DNA. So we want to get rid of all the RNA. We want to get the cells, bust them open, get rid of the RNA. So the first step is get the cells. That's what we're doing right now. Anybody been timing? What are we at right now? We at like 2 minutes yet? Alright, so what we can do is just check. Let's look at our cells. Oh, see, look, we're pretty good. You see how the sample is now clear? And you can see that pellet, that cell pellet right there? This is what it should look like. You should have a cell pellet. That's good. Can we focus on that? Cell phone camera, can you focus on that? Kind of. See, there's a cell pellet and the solution is clear. That's what it should look like. Clear. All right. So now what we're going to do is we're going to dump out the solution that the cells were in. So you can use pretty much any container. It doesn't really matter. So we're just going to use an empty Tupperware container. And you're just going to dump out the liquid that's in there. Dump out the liquid. So the liquid should be, yellowish in color still from the LB broth. And Yeah, Vincent. We're trying to delete all the RNA in there. All right. Now the next step is going to be resuspend in 250 microliters of buffer A1. So we're going to set our pipette to 250. We're going to take a pipette tip. We're going to take 250 microliters of A1. You should have this in a 15 ml tube, and add it to the first tube. What we're going to do is we're going to resuspend by pipetting up and down, and then let's make this a little bit larger, maybe like 400. So we're going to resuspend, and then we're going to add it to the next tube and do the same thing. Resuspend it so that all the pellet is off the side. So your tube should have nothing left in it after. It should just be clear and empty, no pellet. And then just keep doing that until all your cells are mixed in. All right. So let's keep one of these tubes. Let's add this to it. So we have, so you want to resuspend all of yours and put them in one tube. Mine, because I use the shaking incubator, it's a little higher concentration. So I know this is really dense and good, so I can stop here, but make sure you add all of your tubes and resuspend them. Now, the next step, after we resuspend our cells with the RNAs, we're going to add a buffer that's going to lyse the cells. Now, this buffer contains detergent, contains SDS, which is in, soaps. It's in soaps that you use. SDS, it's the same thing. I'm going to resuspend 250 microliters, and then we're just going to rotate this tube kind of back and forth until everything is mixed up and good. Now what should happen is we let it sit for a minute, and what should happen is the bacteria in there should start to all lyse, right? So when you use a detergent, it likes to break down lipids, right? When you use soap on your hands, if they're oily, it gets rid of the oil, because the lipid micelles, the lipids interact with the soap micelles, and the soap can just pull it away and wash it away. The same thing happens in bacteria. The SDS mixes in there and starts breaking apart the bacterial cell wall, which is made of lipids. Now, once the bacterial cell wall is broken apart, then the DNA and other stuff gets released. Right? So, we got the RNase in there still, and the RNase is going to be degrading the RNA. We broke apart the cell wall. So, the next step is we have to harvest the DNA. Now, in order to do that, we need to precipitate out all the crap that we don't want. So that's proteins, that is cell debris, the cell wall, and other things. All these different things we want to get out and precipitate out of solution. So what we do is we use this buffer. Now this buffer, if you smell it, buffer N1, it should smell like vinegar or acetic acid. Now, what it does, what this buffer does is it precipitates out the SDS, it precipitates out the proteins and all the other things, so that what you have left is just a solution that basically contains DNA. So your solution should become somewhat viscous. You can see how there's like a little string of bacteria between the tube and the lid. So that's just all the cellular debris and materials all viscous. So here we add buffer in 1. And it should start to become cloudy. You can see the cloudiness that's starting to form. And then you got to mix it up really good. Really good. You might even want to shake it up. Get it all shaken up. There we go. We got it mixed up nice and good. Now we're going to put this in the centrifuge, and we need to centrifuge it down for about 10 minutes. But remember, like I said, what you want is you want to balance. So you want to take another tube that has about the same amount of liquid in it in water, put it in across on the centrifuge as the balance, and then you're ready to centrifuge. So we're going to centrifuge this about 10 minutes or until all the white stuff has centrifuged down to the bottom. So what we want left is a clear solution. All right. Any questions so far? Is everything pretty good? I'm just going to sit here. See, that's why I said you need to bring a beer, because there's some downtime. So you're just going to chill out and hang out. And you're going to need something else to do. Now, this is probably the longest part. So generally, a mini prep, yeah, there's Plasmid maps. If you go to the Google Classroom, under week 1, there should be a Plasmid map for the GFP and the P-Dusk, P-Dawn Plasmid map. Yes, the bacteria are already transformed. They already have the plasmid in it. So you want to grow them up with antibiotic selection. So it's one thing I forgot to add. When you make the LV broth to grow up the bacteria, you want to add antibiotics to make sure that they have the plasmid. No, they're already transformed. You should have received stabs. You should have received bacterial stabs if you received your kit already. And the bacterial stabs should be the ones that you grow up. So you should have 3. P-Dusk, P-Dawn, and GFP. So grow those up. Generally what I do is I probably start out with 1 to try to get it down, to make sure you have it correct, instead of doing all 3 at once. This helps a lot because if you mess up, then you don't mess up all 3, you only mess up 1. So it makes it, you know, less big of a failure. But yeah, try starting with 1. But yeah, this procedure works for generally any plasmid and any bacteria. So if you transform bacteria or you get transformed bacteria, you can do this. You can grow up the bacteria with antibiotics to select for the plasmid, and then spin it down and use a mini prep kit to purify it. Now, mini prep kits, they're not that expensive. Generally, it's like less than $1 a prep. So you can buy a 50 prep kit on eBay for around 40 bucks. Yeah. So if you're looking for some more a kit or some more materials to be able to do lots more mini preps, check out eBay. Search mini prep and DNA, and you should be able to find mini prep kits that are really cheap. Don't let people scam you into buying expensive ones. They should be about 40 or 50 bucks a kit. Snap. If you get a plasmid and you're trying to amplify it, first you have to transform it into bacteria. So if you get a little aliquot of plasmid, first what you need to do is you need to get some DH5 bacteria, DH5 alpha bacteria, and transform the plasmid into it, or another similar strain. And then you can follow the same protocol. Same if you got like a piece of paper. What you have to do is just put the piece of paper in a tube with some water, so some of the plasmid will come off the paper into the water, and then take that water and transform it. Same thing. And you should get bacteria then that has the plasmid, grow it up with antibiotics, and then you should be able to purify it. It's still going through San Francisco. Well, sorry about that, Mark. Hopefully your kit arrives soon. Let me know if it doesn't, and we'll figure out what's going on. All right. Again, I don't know what time we started. But let's give it like at least another minute. And we'll see. So usually this is the longest step. You're going to have to spin it for a while. Don't try to get around it. Just spin it. Make sure you do it correctly. You don't want a bunch of junk to gunk up your column. So let's check and see what we got. So it looks like it's okay, but not that good. Sometimes this happens where it doesn't pellet down really well, and you just have to shake it up a bit and spin it down again. You see how there's like some water or liquid at the bottom? That sucks. It's not forming airtight. All right, so we're going to try to spin this down a little bit again. Let's give it one more shot. Tom, the beer is, yes, it's Longboard Golden Ale. Big Wave. From Kona Brewing Company. I like Longboard and Big Wave. They're both two really good beers, I think. And my other beer go-to during the summer is 21st Amendment Hell or High Watermelon. Doesn't really taste the watermelon that much to me. I can't really taste the watermelon, to be honest with you. I think it's just like a really nice wheat beer. Yeah, I just like it as a wheat beer. It's pretty good. It's also hot up here. I'm probably sweating. Whew. Alright, so in the meantime, what we can do is we can talk about the column. So this is a mini prep column. It has a collection tube, and it has the actual column. So what you should see is that white thing at the bottom. That's actually what is quote-unquote the column. Now what that is it is generally silica, and silica has a positive charge, and so will bind DNA, because DNA has a negative charge. These columns are really cheap to make. Some people make similar things themselves. You can buy them from China really inexpensively. But yeah, it's a pretty simple experiment. What you're doing is you're making the DNA really negatively charged, so it binds to the column, and then what you're doing is you're neutralizing the DNA charge so that it falls off the column. That's, in essence, the mini prep kit. So after we bust open the cell and we have this little liquid, right, with all the DNA in it, what we do then is we place the liquid over the silica column. The silica column, what should happen is that it should bind the DNA. We wash it to remove any junk that's in there. So we wash using ethanol. DNA doesn't like ethanol. So your wash buffer should be, I think it's like 70 to 90% ethanol. And the DNA doesn't like the ethanol, so it should stay on there. And then we should elute it. So one of the steps that I missed is we need to use, before we add the DNA to the column, we add a column buffer. Now a column buffer, what it does is it just gets the column ready to bind DNA by giving the column the correct pH, right? So it's just kind of altering the pH a little bit of the column so that it's ready to bind the DNA. So what we're going to do, we're going to, I think it's add 100 microliters of the column buffer to the column and let it sit, and we'll spin it out. So generally, if you're adding smaller amounts of liquid, maybe like 50 microliters, you want to try to add it right on top of the column. So if you look in the column, you should see that there's plastic ring around the outside, and then there's a column in the middle. So generally, what you want to try to do is put your pipette tip right above that middle part and drop the liquid on there. All right. With also, what we can do is we will make a balance. So we'll use 100 microliters to balance the thing. Facial hair does not really fit my style. Well, I don't care. Alright. Let's see what happens now. Alright, we're getting it to pellet down much more, but we're going to need to spin it a little bit longer. I had so many cells in there. So let me also spin down this column at the same time. It's hard to keep facial hair off my face, you know? That's the problem. I'm not really big on shaving every day. I'm pretty lazy, so, you know, it is what it is. I try to keep it trimmed at least, otherwise it gets all itchy. Thanks, Kent. Thanks for keeping track. Yeah. Generally, these things, you want to spin them for a certain amount of time, but you also just want to spin them until they're done also. So that's the most important thing, is to wait until they're done. Especially for this step, you want to try to get as much of the white stuff out as you possibly can. So that's important. Spin it till it's done. open a window, let some fresh breeze in here. It feels good. I'm running out of beer faster than I thought. Is there an open source Snap-Gene alternative? I don't know. Snap-Gene Viewer is free though, so you could use it for free. If you want to actually use the real Snap-Gene, I'm sure there's stuff out there. But yeah, we'll go through editing DNA and stuff like that, designing it later. Right. So it's been about 3 minutes. Let's wait another minute or two, and then we'll stop it again, see how it's doing. So usually when there's a lot of white stuff like that, it means you use too many cells. So if you grew up, say, 10 ml, and you use all 10 ml in your mini prep, and you got a lot of white stuff like this, it usually means that you had a lot of cells. So next time, cut down on the amount of cells you grow up and use. So maybe use 5 ml the next time. Oh, Tom, that bacteriophage-associated origin is just like a leftover. When people used to do stuff with bacteriophages and other stuff, like, it's not needed. You have to understand, like, when you look at a plasmid map, there's a lot of stuff in a plasmid map that's not needed or is just like a leftover or some other junk. So, don't think that because it's in a plasmid, it's there for a reason. Don't think that at all, because that is not always the case. Plasmids have been around for a long time, and 20, 30 years ago, it was really hard to stitch things together. So when people did it, it was important, and people used it. Nowadays, if somebody gives you a plasmid, and they say use this one, and it has extra origins and stuff, you're just like, I'll just use another one. So it's just that the plasmid is derived from an older series of plasmids. Alright. So, first our column, you should see a little bit of water at the bottom of the column. That should be our buffer that went through. Let's take out the balance. And let's see how our white stuff is doing. It's going down even more. Let's see how much of that white liquid we can get out, so we don't have to spin it anymore. So what I'm going to do is I'm going to use my pipette very carefully and try to just suck out the liquid that is. I'm going to add it to my mini prep column. You got to be really careful if you do this, that you don't suck out any of the white stuff, because what the white stuff will do, it'll just clog your column, and you don't want that. Alright. So put that back in. And what we could do is we could centrifuge down this thing. So let's add a little bit more liquid to our balance, so that it's closer. We'll put it back in. We'll put them all back in, see if we can get the white stuff centrifuged down a little bit more. If not, not working. So mostly stuff should move through the column pretty fast. You should only have to spin stuff for about 2 or 3 minutes from now on. If you have to spin it for longer, if stuff's not moving through the column that well, it probably means you clogged your column. There's too much cell debris and other junk that you added on top of your column. So you got to be careful when you are transferring the liquid away from the cell debris to make sure you don't add a bunch of junk, right? Don't add junk. Not at all. Try to get just the liquid. That's where the DNA will sit. That's all you should be worried about. All right. So let's give this about 2 to 3 minutes, and then we'll move on to the next step. So the next step, what we're going to do is we are going to wash the column. Like I told you before, so there's going to be a lot of junk after this. There wasn't just DNA in there, there was some other ions and other junk that, probably stuck to the silica with the DNA. So what we're going to want to do is we're going to want to wash the column to remove all that stuff, but keep the DNA. And generally that's done with an ethanol solution. So that's what we're going to do next. So you see we got all the liquid that came through the column. We got any more liquid on top of this white stuff? A little bit. So let's add a little bit more to our column. Now it's trying to suck up the white stuff. I don't know if it's going to work. Yeah, we'll leave it. I think we're good. Alright, so you want to empty out, decant, they call it. You just dump it out, whatever goes through the column into the waste. And then we're going to add 700 microliters of wash. So add it directly onto the column. Make sure you have a balance in there. Put it in, and let's spin about 2 or 3 minutes. All right, so we're almost done with this. After this, what we want to do is we want to spin it again for another 2 or 3 minutes. Or this last one is important, so you could spin it for 5 minutes, I would say. I tend to spin it a little bit longer than normal. What you're trying to do is you're trying to get rid of any residual wash buffer, because residual wash buffer is going to contaminate your mini prep. So you're going to want to spin it for a little while longer probably than what people tell you or what guidebooks tell you, just in case. So if it says like 1 minute, 5 minutes is generally a good number. You can't over centrifuge these things. You can't over centrifuge it. So it's okay to spin it for longer, because residual wash buffer is not a good thing. Now, after we spin it down the final time, what we're going to use is an elution buffer. Snap, yes, you need antibiotic in the LB broth. The antibiotics should be in the Plasmid maps, maybe in the guides, but the GFP plasmid should be ampicillin, and the RFP pidone plasmid should be kanamycin. And you can look up working concentrations online. If you're not sure what concentration of antibiotic you should use with bacteria, just search on the internet working concentration ampicillin, working concentration kanamycin, and they should give you a working concentration. Now the working concentration is just the final concentration that you want. Generally, you want to make a stock solution of the antibiotic that's about 1,000 times stronger. So then you add 1 microliter per milliliter of media. So ampicillin is generally 100 micrograms per milliliter, final working concentration, but you want to make a stock at 100 milligrams per milliliter. So if you're making 5 milliliters of ampicillin stock solution for your media, that would be 0.5 grams or 500 milligrams for 5 mLs. Now, not all antibiotics are soluble in water, but kanamycin and ampicillin should be. Use distilled water if possible. Alright, so let's take these out. Alright, so you see we got all the liquid, so let's decant that. And now let's spin it again. Like I was saying, we want to spin this one more time. We need to find something for the balance, something lighter. So we're going to spin it one more time. We're going to try to get rid of all the residual wash buffer. Now there's one more thing we need. I need a clean microcentrifuge tube, which it doesn't look like I have. So I'm going to run and get one. Let me see. Alright. Alright, so I got a clean tube. So what we're going to do, like I said, let's let this spin for a little bit. We're not concerned about overspinning, we're more concerned about underspinning. Yeah. Want to get rid of all that residual wash buffer as much as we can, because it's bad. Alright, so the final step, what we're going to do is we're going to add elution buffer. Elution buffer can basically just be water, just normal pH 7 water. Don't use tap water or anything like that. Generally you want to use, some special water or some elution buffer, but generally distilled water will work. But we should have sent you some elution buffer, so use that. Elution buffer is generally TE, which is just tris EDTA. EDTA helps chelate ions so that DNases and things that break down DNA can't work as well. So It's good for long-term storage of DNA. Sometimes TE can inhibit other downstream experiments, so water is also, sometimes better to use and is great to use in a pinch, but the elution buffer should work fine. All right, let's check it. Now, you should have some more liquid in the bottom of your tube. If you don't have liquid in the bottom of your tube, it means you didn't spin it long enough. And there's probably still some residual wash buffer in your column. So spin it again. But yeah, you should have some liquid in the bottom of your tube. So then what we do is we take our column and we put it in a fresh microcentrifuge tube. Don't forget that. I forgot that before, and what ends up happening is your DNA prep is gone because it gets mixed in with all the crap that was just in the bottom of that tube. I don't know. So generally, it says you can elute with, different amounts. You can always elute with less. It's generally always better to elute with less and not more, because if you elute with less, your DNA is more concentrated. It's better to have more concentrated DNA, which you could then dilute, than to have less concentrated DNA, which is hard to concentrate. Because usually when you concentrate it, you lose a lot of the DNA, right? So I generally, in a mini prep like this, you're going to want to add 50 microliters of DNA. 50 microliters of elution buffer. If you think that you probably didn't have that many cells and you want to do less, you can probably get away with like 35. But yeah, be careful, because once you start getting lower and lower, you might not elute everything off the column. So the lowest that probably goes 35. Generally, for a mini prep like this, you don't want to go more than 50. So let me get the elution buffer. And remember, you want to place, because it's such a small amount, you want to place it directly on top of the column, above the white spot. So you want to look in there really close, make sure your pipette is right above the white spot, and then press and watch the drops go into the column. Tap it down, make sure everything's in there. And good. All right. Now what you want to do is you want to give the elution buffer a little bit of time to work. So we're just going to let it sit for like a minute or two. There are certain things that help DNA elute, but be careful when you use these. Use them only when you're desperate. One thing is warming up the tube. So if you warm it up to say like 37 degrees, you generally will get more DNA eluted off, but that also means sometimes you'll get some junk with it. So be careful, because it means that your prep is going to be dirtier, but you might have a higher quantity of DNA. All right. Any questions before we do our last spin? So one thing I forgot to mention that is always important, if you're doing multiples of these, you should, number one scientific investment is these thin Sharpie markers, ultra fine point Sharpie markers. Buy some of these because you need to write on these microcentrifuge tubes. So buy some ultra fine point Sharpie markers if you don't have any. And you want to label your tubes, right? So in this case, if you're doing GFP, you can just put G or GFP. You want to label your column, and you also want to label the top of your tube, because that's the important part. And you don't want to mix up your tubes, right? So make sure you always label often your tubes. All right. So we're going to spin this down and stick it in there just like that, and then close it up. and let it spin for, 2 to 3 minutes. All right. Basically, that is the gist of a mini prep for plasmid DNA purification. Now, obviously, like anything else, you're going to have to try to get the hang of it. It might take a try or two. So, generally start with one plasmid, like I said, see if that works. If it doesn't work, try it again, or try one of the other ones, but do them one at a time. When you grow up the bacteria and spin it down, you can always freeze the DNA, freeze the bacterial pellets. So what I do a lot of times is I'll just grow up bacteria, spin it down, and stick them in the freezer and save it for future, when I want to purify it in the future. You don't have to do it right away, so don't worry about that. All right, let's check this out. So generally what you're going to get is less than 50 microliters. That's because you're not going to be able to spin all the DNA off the column. So if you get less than 50 microliters, don't freak out. Understand that it's always going to happen. I've never gotten, 50 put 50 microliters on and get 50 microliters out. If you get more than 50 microliters, that means you got excess wash buffer, right? And that is not a good sign. Sometimes if it's not too much more than 50, you're going to be okay. But if it's a lot more than 50, if you get, say, 100 instead of just 50, your DNA is probably going to be pretty shit. So I would just start over. Tom, how long can you freeze the pellets for? You can freeze them indefinitely. You can freeze them for 10 years if you want to, generally, and they should be okay. So there really is no time frame. Just freeze them after you spin them down, because if you freeze them while they're still in the liquid culture, so freezing is also good because what it does is the ice crystals break apart the cells. So when things freeze, you make these ice crystals, and the ice crystals will stab into the cell walls, and they'll break apart cells. But what happens if they're not pelleted, all that DNA and everything that gets broken apart in the cell gets released into the solution. So just make sure you pellet the bacteria before you freeze it. Don't resuspend it in A1 buffer or anything. Just pellet it, put it in the freezer. No, you don't freeze with the supernatant, Mark. Don't freeze with the supernatant. Pellet, dump out all the liquid, so you just have the cell pellet. You should only have the cell pellet. That's it. As little liquid as possible. Because, you're eventually going to remove that liquid or whatever, and that liquid you don't want it to contain the DNA that you want. So freeze the cell pellet, dump out the supernatant, put it in the freezer. All right. Any questions on this class today? So the next thing you're going to want to do after you purify your plasmid DNA is you're going to want to run it out on a gel. I don't know if that's experiment is this week or next week. I forgot. I guess I could check really quick. Let's see. What do we have this week? So we have growing up bacteria, and next week we have running an agarose gel and analyzing. So if you purify your plasmid DNA and you have time and want to skip ahead, you can run it out on an agarose gel, but we'll talk about that next week. We'll go over how to analyze an agarose gel and look at it and what the different results and stuff are. How do I print the small labels? We buy specific labels and stuff like that. Generally, you can write on the tubes. So the top of the tubes and the side of the tubes are made so you can write on them. So the top of the tubes have like a certain texture, and you could see on the side of the tubes where there's a little bit of texture. You can write on that pretty easy with a Sharpie marker. So get some of these ultra fine tip Sharpies and ultra fine point. Be careful though, because things like ethanol from the wash buffer can wipe off markings. But using the permanent marker, they should stay put and last a long time. So good thing is with all your tubes, whenever you're doing an experiment like this, whenever you purify DNA or put bacterial pellet in the freezer, label it. So write what it is and write the date on it, right? So this is GFP, bacterial GFP that I purified, and I purified it on September 10th, right? Because, you know, you're gonna want to know at one point or another. It's not a dumb question mark at all. You generally want to store everything in the freezer. All DNA and all DNA things you want to store in the freezer. If it's like bacteria, like a stab, store it in the fridge. But anything that you're trying to preserve over the long term, store in the freezer, except for living organisms, right? Because living organisms don't like to be frozen mostly. So anything that's dead, store in the freezer. Any other questions before I go? Again, if you have any questions, feel free to contact me. Feel free to post in the Google Classroom, and I'll do my best to answer them. We also have a Facebook group for the ODIN classes. Join that, and in the ODIN Facebook group, other people who've taken the class previously will help you, give advice and other things like that. All right. So thank everybody for coming this week. I appreciate it. If you have any questions, let me know. Otherwise, I will see you next week, and hopefully you'll have your experiments done.